physiological processes altered by muscles. Virtually all of our dynamic interactions with our environment depend on our ability to move, depend on our muscles. Skeletal muscles. Remember, there were 3 types of muscles we talked about. We talked about skeletal muscles, cardiac muscles, and smooth muscles. We showed you a histological slide. You should identify each one of those from the histological slide. So skeletal muscles are voluntary muscles. Okay, they're elongated cell fiber. They're elongated cells called fibers. So the cells are stretched out and they're long, and they're called fibers, muscle fibers. Fibers contract along their longitudinal axis. That's all the muscle can do is contract. Can't do anything else. Contracts and relaxes. It has to contract along the longitudinal axis. It pulls together. It can't pull that at different angles or anything like that. It has to contract at that. 3 types of muscles we talked about. Skeletal muscles, cardiac, and smooth. Skeletal muscles are voluntary. We can control that. If you want to move your arm, that's skeletal muscles. If you want to look in the corner of the room, those are skeletal muscles. Okay? It pulls on skeletal bones and forces movement. Cardiac muscles. Cardiac muscles are involuntary. It pushes blood through arteries and veins. Okay? Has rhythmic contractions. Okay? If you're looking at a histological slide of a cardiac muscle, what is the key thing you're going to look for that is going to differentiate the cardiac muscle from skeletal muscle?
Intercalated discs.
Perfect. Intercalated discs. The ability of all the muscles to contract at the exact same time so the heart pumps blood efficiently. You look for intercalated discs. Smooth muscle. Smooth muscle pushes fluid and solids along the digestive tract, and it's involuntary contraction. So skeletal muscles and cardiac muscles have striations when you look at them on the histological slide. Smooth muscles do not. Okay, so muscle tissues for 4 basic properties. Every type of muscle fiber. Okay, excitability, the ability to respond to a stimulus. Okay? It gets excited. There's a stimulus, and the muscle can respond. Contractility, it shortens and exerts a pull or tension. So muscles have to be able to shorten. That's how they exert their effect. Okay. Extensibility, the ability to continue to contract over a range of resting length. In other words, if you're going to grab the bottle to take a drink out of, you don't crush it all the time because your muscles can contract to a certain limit. You have the ability to control a range of resting movements. They're going to contract till you can grip it, and they're not going to contract till you squeeze it and bust it in half. Okay? So you have that range of muscle, of strength. Okay? Elasticity. In other words, once the muscle contracts, it has to relax. If it can't go back to where it was, it does you no good. So it has to have excitability, contractility, extensibility, and elasticity for the muscle to function. All 3 types share those 4 basic properties. Skeletal muscles. Skeletal muscles are voluntary muscles. They produce skeletal movements. They pull on tendons to move bones. Remember, a tendon connects a bone to a muscle. A ligament connects a bone to bone, and aponeuroses connect muscle to muscle. So if you're pulling on, if a muscle's pulling a bone to move it, it is a tendon, because tendons connect muscle to bone. Okay, that's how you produce skeletal movement. It maintains body posture and body position. If you're sitting here listening to me, you're not laying down, you're standing up. It's the muscles that are controlling your posture and your position that allow you to do that. Allows you to head to stay on top of your and not rotate. If you're in class, I watch people just kind of their head would slap to the side because they'd fall asleep, and you lose that muscle tone. And it supports soft tissue, supports the weight of all your visceral organs. It supports your heart, your lungs, your stomach, intestines, and everything there. Okay? Those are the functions of skeletal muscles. Okay? They also regulate entering and exiting of material. So voluntary control for swallowing, defecation, and urination. Okay? That's under voluntary control. You can control that. Okay? I deal with a lot of child abuse. I do forensics. I still do a lot of child abuse. And one of the things you look for is a constipation in a kid. They won't defecate. They don't do it because they're in an environment where they can't control what's happening to them. And this is something they can control. So a lot of kids that are constipated backed up like that are voluntarily creating that because it's something they can control. It gives them something in their environment that they have control over, because everything else is out of control for them.
Would it be, is that like subconsciously, or is that like purposeful?
It's hard to say, but we see it a lot with abused kids. It's probably at a subconscious level, but they have to have something in their lives that they can control. Well, just like you've got to control things, right? You've got to be in control of things in your life. And they have no control over what's happening to them. So they control by not defecating. So it's probably a combination of the two. It's real interesting when you deal with kids like that. Like I said, I have a whole lecture on child abuse and the John Benet Ramsey case. That happened to that. Maintain body temperature. Energy is used for contraction. When you shiver, you convert that to heat. Okay, so you regulate what comes in your body, what goes out of your body, and helps to maintain body temperature. Those are the functions of skeletal muscles. Okay, so gross anatomy. Gross's overall anatomy is the organization of the muscles. Connective tissue associated with those muscles, nerves associated with the muscles, and blood vessels associated with the muscles. That's your overall gross picture of anatomy of the muscle system, of your skeletal muscles. So their organization, the connective tissue, nerves, and blood vessels. How everything's connected. Microscopic anatomy is the study of the myofibrils, myofilaments, and the sarcomeres. Sarcomeres is the unit of a muscle cell. We'll talk about myofilaments and myofibrils in just a little bit here. All right, so connective tissue of the muscle has 3 layers. There's an epimysium, there's a perimysium, and there's an endomysium. The epimysium is the outside layer. It's a dense tissue that surrounds your entire muscle cell. That's your outer layer, your epimysium. Your perimysium, that's the dense tissue that divides the muscle into components. There's different parallel components that can contract individually inside a muscle. That's your perimysium. That's the division. Then the dense tissue that surrounds each individual muscle fibers is the endomysium. So you have the epimysium on the outside, the perimysium divides into the endomysium that divides into each individual muscle fiber or cell. And that's what it looks like. This is your epimysium right here. This is your outer coating. Okay, that's your endomysium. Your perimysium is divided into each of these individual cells here. This is your perimysium. Goes around each of these individual groups. So endomysium is the outside, then the perimysium, and then the endomysium goes around each cell inside of that. Okay, so epimysium, perimysium, endomysium. You can see there's a better picture right here on the top. Again, the outer layer, then the layer that divides each one, the way it divides inside of each one. Okay. Okay, with the connective tissue muscles, you have the epimysium, perimysium, endomysium, and all those converge to form tendons. Remember, tendons connect muscle to bone. Aponeuroses connect muscle to muscle. And ligaments connect ligaments connect bone to bone. Ligaments are bone to bone, tendons are muscle to bone, aponeuroses are muscle to muscle. All right, so nerves and blood vessels. Nerves innervate muscles by penetrating the outer layer and enter into the muscle fibers. There's a chemical communication between a nerve and a muscle. That's your neuromuscular junction. We talked about that. There's a question on ACH last time, acetylcholine. This is where this is going to come in. This is the chemical that's released into the neuromuscular synapse. The neuromuscular is a space. The nerve comes. This is your nerve cell. I'm not great at nerve cells. And this is your muscle fiber right here. Here's your striations. And this nerve, okay, this is your neuromuscular junction. That's where the nerve and the muscle meet. Okay? That's how it innervates, it ends. So when the signal comes down this nerve for the nerve to fire has an electrical impulse. It releases chemicals into this cleft. That chemical released is ACH, acetylcholine. Okay? The acetylcholine travels across the synapse, the space, binds to this muscle cell, causes the release of calcium into the joint. Calcium combines with ATP, causes it to contract. And we'll go over all that. Okay? But that's the chemical involved at the neuromuscular junction. That's how muscle contracts. It gets a signal from a nerve, releases what's called a neurotransmitter, which is ACH, it's chemical into that synapse. Okay? Blood vessels often parallel nerves that innervate the muscle. Okay? So where you have nerves, you'll have an artery and vein running together. That's why nurses aspirate when they're doing blocks. You don't want to inject the anesthetic into a blood vessel. You want to leave it locally in the area you're working at. Okay? So along the nerves, you want to get a nerve numb, do a nerve block. You have to remember blood vessels run right alongside that nerve. So everybody know what aspirate means? To pull back on a syringe. Make sure the tip of that needle is not in a blood vessel. Because if you got nerves, you got blood vessels. This is blood vessels here. This is your veins running here. When you put a needle into there, you want to go right at that nerve. You don't want to go into a blood vessel. You don't want to inject your stuff into blood vessels. One thing, it's going to travel away from the site, and it's not going to work. Another thing, it could have serious side effects. It is epinephrine, like what we use epinephrine in ours to keep it localized. And that's not good for the heart in certain patients. So blood vessels parallel nerves that innervate muscles. You're going to find them running together. They then branch to form coil networks to accommodate flexing and extension of muscles. Remember, the muscles that cause flexing and the muscles that cause extension are opposite to each other. There's going to be different innervation to each one. The body's going to have to signal flexing or signal extension, and different muscles will be activated to shorten. Okay? If you flex and extend, it's not some muscles contracting to make them shorter, it's somebody making it longer. Okay? It's different muscle groups. They're both contracting to become shorter. Okay? Certain muscles flex, certain muscles extend. Okay? The skeletal muscle fiber, the sarcolemma. Remember the cell, we talked about the plasmalemma. That was the cell membrane. The sarcolemma is just a membrane around a muscle cell. Sarcoplasm is the cytoplasm inside a muscle cell. It's just like cytoplasm, except the muscle consists of sarcoplasm. And the muscle fiber, we talked about muscle fibers, is the same thing as a muscle cell. They can be 30 to 40 centimeters in length. They can be really long. They can be multinucleated. So each muscle cell has hundreds of nuclei for skeletal muscles now. We're talking about skeletal muscles. Nuclei located just deep to the cell membrane, the sarcolemma. Okay? So they can be long. A muscle fiber is a muscle cell. Move that back. Okay. So muscle fibers develop through the fusion of the mesodermal cells called myoblasts. These are your myoblasts. They form myosatellite cells, and then you get an immature muscle fiber, and this can turns into a mature muscle fiber. That's how they develop. So myoblasts develop fibers. Once the muscles are formed in your body, you're never going to get any more. Muscles don't repair themselves. Okay? If you pull a muscle fiber, it's going to stay pulled the rest of your life. Now, you can have certain repairs, like when athletes pull a hamstring, the muscle is going to tear. Okay? That tear is going to be different. What happens is you have blood, and it bleeds, and it forms a scab. That scab can hold that muscle together to a certain extent. Okay? But muscles do not regenerate. You do not grow new muscles. You're born with as many as you're going to have. You can make them larger and stronger, but you're not going to make more muscle cells after birth. Myofibrils and myofilaments. So the sarcoplasm contains myofibrils. These myofibrils are responsible for contraction of the muscle. There's going to be thick ones called myosin, thin ones called actin. These are the fibrils that move past each other and cause the muscle to shrink, okay, or shorten or contract. Okay? The myofibers are attached to the sarcolemma at each end of the muscle cells. So if the fibers are attached at each end, as they pull together, what does that do to the ends of the muscle cells? It pulls them closer together, causing contraction. So surrounding each myofibril is a sarcoplasmic reticulum. Remember we talked about an endoplasmic reticulum. This is a sarcoplasmic reticulum. The sarcoplasmic reticulum's job is to release calcium into the cleft. That's what causes muscles to contract. The presence of calcium. Muscles will not contract without the presence of calcium. It takes two things. It takes calcium and it takes ATP. That's a P. Okay. You have to have calcium and ATP to let the muscle contract. Okay. The myofilaments, like I said, the thin ones are actin. The thick microfilaments are myosin. And they slide past one another. And as they slide past one another, they pull the ends of the plasmalemma together, shortening the muscle. And we'll go into great detail how that works. So myosin is your thick filament, actin is your thin filament. Both are arranged repeating units called sarcomeres. Those are repeating units. So they all we can contract as units. Okay? All the myofilaments are arranged parallel to the long axis of the cell. You don't have them arranged this way, this way, this way. They're all arranged in one direction. So when they pull, they're always going to shorten along the long axis of the muscle fibers. Okay? You're always going to be along the long axis, and they're always going to shorten for skeletal muscles. Okay? Sarcomere, remember we talked about that's your main functioning unit of a muscle cell. Okay? There's about 10,000 per myofibril. So every myofibril has about 10,000 sarcomeres, 10,000 units it's going to pull on. That consists of overlapping actin and myosin. That's what allows us to slide past one another. Overlapping creates striations that gives the skeletal muscle the striations that we see histologically. So it's the overlapping of this actin and myosin that you see in the striations of the muscle cells. So in the sarcomere organization, skeletal muscles consist of muscle fascicles. Muscle fascicles consist of muscle fibers. Muscle fibers consist of myofibrils. Myofibrils consist of sarcomeres. Sarcomeres consist of myofilaments. Myofilaments are your actin and myosin. Your actin and myosin causes contraction and the ends of the sarcolemma to pull together and shorten the muscle. That's basically what they're made up of. That's their whole organization right there in a nutshell. Any questions so far? We all good? Cool. All right. So a contracting muscle, remember when a muscle contracts, every single cell, every fiber is lined up in the same parallel direction. They have to shorten the muscle when they contract. That's all it can do. Can't do anything else. So a contracting muscle, it shortens in length. No exceptions. Okay? Contraction is caused by interactions between thick and thin filaments, the actin and the myosin. We'll show a video in just a second on how the contraction, which is actually really well done. Contraction is triggered by the presence of calcium ions. Remember that you have to have calcium ions released by your sarcoplasmic reticulum and ATP for muscle to contract. Okay? So muscle contractions require all the preponderance of ATP. So those two things, ATP and calcium. When a muscle contracts, actin and filaments slide towards each other. The sliding action is called the sliding filament theory, where myosin and actin slide past one another. Okay? So we talked about this a little bit at a synaptic junction. A neuron sends a message, your brain sends a message, an electrical impulse. This electrical impulse travels down the nerve, the axon. At the end of the axon, where it meets the muscle, acetylcholine, ACH, is released. That goes into that synapse we talked about. Okay? From the end of the axon into the neuromuscular synapse. That's where the ACH is released. This causes a sarcoplasmic reticulum, okay, to release the stored calcium ions. So the sarcoplasmic reticulum then releases calcium into the synapse. That starts the actual contraction. So it's not the nerve impulse that causes that's directly to it. The nerve impulse at the end stimulates the sarcoplasmic reticulum, which releases calcium into that joint, into that synapse. Calcium, the presence of ATP, causes the muscle to contract. Okay? So this is your nerve cell here. This is your axon coming down and it innervates over the top of the muscle. Once it gets that signal, okay, you get your synaptic cleft and it releases calcium from the sarcoplasmic reticulum and causes contraction. See, so you have at the end of each nerve, you have these little vesicles. Inside these vesicles is ACH. Once it's activated, this ACH is dumped. Oh, I made that arrow too far. Okay, so the ACH goes right into the cleft. This is the cleft right here. So this is the nerve cell, and this is the muscle cell. Okay, and these are little receptors. So the ACH binds receptors causes the calcium to be released. All right, let's watch a short video here. It's actually a really good one, the sliding filament thing.
You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed.
Oops. Was that showing the pictures change? Was that showing the video?
It was.
Or not? They just have one picture.
It
You want to show the video? Oh, okay.
You use muscles every day to do activities. This woman is using muscles to breathe, circulate blood, and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate, causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When the muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
That's a real good video on how that actually works. And when you think about it, remember the muscle contracts and it shortens. Okay? They're locked together. ATP allows that bridge to disconnect. So think about it. When a patient dies, rigor mortis sets in, right? So the muscles are contracted because the body stops making ATP once the person dies. So no ATP is around, so those bridges are locked. You can't have ATP to release them. That's what rigor mortis is. The muscles contract, and the ATP is not there to let that release anymore. That's how come rigor mortis sets in after a certain time frame following death. But that is a real good video. I enjoyed that. That really explains it well. So muscle contraction, to sum up that video, a nerve impulse, okay, causes a release of the neurotransmitter acetylcholine, which comes in contact with the sarcoplasmic reticulum. Okay? Neurotransmitter causes the sarcoplasmic reticulum to release its stored calcium ions. Calcium ions bind to troponin. Okay? And that begins your contraction. So there's your calcium ion. So the contraction is a series of all these steps. So the contraction begins when calcium is released into the cleft. Okay? Calcium binds to the tropomyosin here and exposes the active site so you can get binding. So this can connect. Okay? So the bound calcium ions cause the tropomyosin molecule to roll to the side, so it exposes the active sites on the actin. That allows them to bind together. And it's like a ratchet that pulls across. The myosin head extends and binds to the exposed site and pulls across. So the actin and the myosin slide past one another. Once the myosin head binds to the active sites, they pivot in the direction of the M line and shorten the M line. So the cross bridge forms. Okay. So once the active sites are exposed, the myosin heads, which are these right here, okay, of adjacent thick filaments bind to them, forming a cross bridge. And then these pull in this direction. The myosin heads pivot and pull the actin along towards the M line. So upon pivoting the myosin heads, the actin filaments slide, excuse me, slide towards the M line. The sarcoplasm at the end of each cell starts to pull together and shorten the cell. Okay. ATP binds to the myosin head and causes them to release their attachment and return to the original position to start to make another cross bridge. So if ATP is not present, the muscle will start to contract and pull, and will have no way for the myosin head to release the actin. Okay, so when ATP gets used up, like I said, when you die, okay, muscles go through rigor mortis. They contract and they can't release because the ATP isn't there because you're not producing anymore to allow the myosin to release from the actin. Okay, and here's your cross bridge. ATP allows this to release, swings back, forms a new one, and pivots. Releases, forms back, and pivots. And that's how it's like a ratchet motion. Keeps moving forward, 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 and keeps sliding inch by inch by inch. Okay? So when you contract a muscle, the I bands get smaller, the M lines move together toward each other, the H bands get smaller and the Z lines get closer together. Okay, so you're contracting that muscle. Succinylcholine. Succinylcholine used to be used in operating rooms. Succinylcholine is what's called a competitive inhibitor of acetylcholine. Okay, it's used as a muscle relaxant in surgical procedures. Now, there's some dangerous properties with that when you think about how this works. A competitive inhibitor occurs because the two molecules are so similar that it binds to the acetylcholine receptor. So whether you have that nerve coming down here, you have your nerve feeding into your muscle cell, okay? And it travels into this joint here. There's receptors that bind the ACH that gets released from the nerve. If there's another molecule that's injected into this site that has the same shape as ACH, acetylcholine, then that's going to bind into the sites for the receptors. And ACH will not be allowed to bind into there. It will not have the same effect. It'll bind them, but it won't do anything. It blocks them, basically. Okay? So when you administer a patient, the more succinylcholine, okay, than ACH in the synapse, more succinylcholine binds to receptors. Receptors become non-functional, and the patient can't move. And you get paralysis, and you can do your surgery. You cannot stimulate muscle contraction. Succinylcholine causes the muscle to relax. You can't contract your muscles because you can't get the ACH to bind the receptors, which stimulates the calcium release in the sarcolemma reticulum. That doesn't happen if succinylcholine is present because it binds the receptors and ties it up. That's called competitive inhibition. They compete with each other. Okay? So normal muscle contraction resumes when the drug is no longer administered. So you stop doing it. Now, you got to be very careful with this stuff. Okay? Remember, the heart's a muscle, the lungs are muscles, everything stops. Okay? Now, if you want to do surgery in the operating room, the person cannot move. Does this do anything for pain? Not a thing. Patient is completely paralyzed, but can feel everything you do to them. There are a lot of horror movies based on that fact. Okay, so when you use it in an operating room, you have to use it with an analgesic, something that causes the pain to stop also. You can't just give them succinylcholine. Succinylcholine paralyzes them, but they can feel everything that goes on. Okay? So you got to be really careful when you're administering drugs and understand how the drugs work and what they do. Okay? You and I have many return patients, I guarantee you, if you only use succinylcholine on them. Okay, you also can have inhibitors. Now, you remember it's released into the joint. Okay, so here's your nerve again, coming down, and it's releasing ACH into this joint. Here's your muscle. Okay, now ACH causes this muscle to contract. Okay, it causes the muscle to shorten. Now, if ACH is present, it'll keep contracting. There's something that has to get rid of that ACH once it's stimulated, right? The muscle has to stimulate, then relax. You can't have the muscle continue to be stimulated. That's tetanus. You can stimulate a muscle so hard, you can actually snap your back. You snap your spinal cord because the muscles contract, 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 and nothing relaxes them. So we have something called ACH esterase, which is ACHE. That destroys the ACH after it works from the joint. So now the muscle can relax. It can stop being activated. If you have a poison or a drug that inhibits this from happening, you can cause your patient to spasm where they can never relax their muscles. And again, they will contract so hard they'll actually snap their spine. The muscles will contract and contract and contract. And these are called, there's some irreversible ones and reversible ones. There's different types of toxic effects, different types of poisons that act like this, and they inhibit that acetylcholine esterase. So you've got to have the ability to put acetylcholine into the synapse, and you've got to have the ability to get it out of the synapse. So the muscle can contract, do its function, and then stop contracting. You've got to have both. And if you inhibit at either end, it's going to cause effects. One will paralyze, one will cause continual contraction until the muscles contract and they can no longer contract no more. All right, motor neurons, okay? Those are motor neurons controlling muscle fibers. You can have precise control or less precise control, okay, depending on where it's located. A motor neuron controlling 2 or 3 muscle fibers, okay, is going to have very precise control. You're controlling 2 or 3 individual cells from one neuron, like your eye muscles. Your eye muscles, tremendous control. You can focus on a word, an individual point. You have specific control of your eye muscles. A lot of innervation in each cell. Same with the tips of your fingers. You can grab things. You can write with a pen or pencil. You can hold on to things. You have dexterity. Okay? You have precise control over that. You have less precise control in like your control of your leg muscles. Okay? Your leg muscles are these massive muscles that hold your body. Okay? Those have less precise control. So you got one neuron and it can control maybe 2,000 muscle fibers because you don't need to control your leg muscles that accurately. Okay? So depending on the innervation, if one neuron innervates 2 or 3 muscles, you have very precise control. If one neuron innervates 1,000 fibers, muscle cells, you won't have precise control. But you'll get contraction, but you won't have that ability to control the contraction to the amount that you need like to write with a pen or pencil or something like that. Okay? So muscle tension, okay, depends on the frequency of stimulation, how often it's stimulated, and the number of motor units involved. Okay? So a typical example is a muscle twitch. If you ever have your muscle twitch on you, it'll shake. Okay? That's the stimulation. It's being stimulated over and over again. The number of units involved, you have complete contraction or no contraction at all. It's an all or none pattern. So when your muscle fiber is stimulated, it contracts. It either contracts or it doesn't. It doesn't contract halfway, it doesn't contract a quarter way, it contracts or it doesn't. Now the number of muscle fibers that contract, you control. That's for the strength of your contraction. How hard you grip someone's hand, how hard you can hold a pencil. You know, how hard you hold on to something when you're gripping to support your weight. Okay? Depends on the number of muscle fibers contracting. Okay? So it's an all or none response. The muscle fiber itself, that cell, either contracts or it doesn't. It doesn't do this halfway stuff. It's all or none. The amount of force depends on the number of motor units that are activated by that neuron. So how many of the muscle cells contract gives you your strength of your contraction. So you're not contracting a muscle halfway. You're contracting it all the way, but you're only using a certain percentage of them if you don't want to have full strength. So muscle tone is the tension of muscle when it's relaxed. That's your muscle tone. Okay? That stabilizes your bones and your joints. That controls the joints to hold their place. So the amount of muscle involvement that results in a normal body position. It stabilizes where your muscles are. Muscle spindles. Muscle spindles are specialized muscle cells that are monitored by sensory nerves to control muscle tone. Okay? Your muscle tone allows your head to balance on your neck when you're sitting here listening to me talk. That's just muscle tone. Those are your muscle spindles. Okay? They monitor sensory nerves to control that muscle tone and allow it to balance it. Muscle hypertrophy. Hypertrophy means to get larger. Okay? Muscle hypertrophy is enlargement of the muscle. Exercise causes muscle. Exercise does not cause you to have more muscle cells. Okay? It does not do that. It increases the size of the muscle, causes hypertrophy. It causes an increase in the number of mitochondria within the muscle cell, causes an increase in activity of the muscle spindles. So the muscle tone is tighter. Okay? There's an increase in the concentration of glycolytic enzymes to break down glucose so more energy is available. There's an increase in your glycogen reserve so the muscle can function longer and fat harder. There's an increase in the number of muscle fibrils, okay? Your actin and myosin, so you can have a more forceful contraction. And there's a net effect is an enlargement of the muscle, what all those things do. Again, you do not increase the number of muscle cells. You increase other factors inside the muscle, and you increase the size of the muscle cell itself, muscle fiber, and you make it stronger. Muscle atrophy. Atrophy is the exact opposite of hypertrophy. Okay, you know the expression, if you don't use it, you lose it? That's atrophy. Okay, it's a discontinued use of a muscle. Causes a decrease in muscle size and a decrease in muscle tone. Okay, you ever have a cast on your arm or your leg and take it off? What's your muscle look like when that comes off? It's really skinny,
smaller
right?
than the other side, yeah.
Exactly, it's a lot smaller on the other side. It's wasting away, it's not as massive. Okay? Physical therapy helps to reduce the effects of atrophy, but atrophy occurs when you do not use a muscle. All right, there's 3 types of muscle fibers, 3 major types. There's fast fibers, there's slow fibers, and there's what's called intermediate fibers. Now you can guess what they do. Fast fibers contract really fast, really quick reactions. Intermediates kind of between the two, and slow. These are long marathon distance muscles. All right? So fast fibers are your white fibers. They're associated with eye muscles where they have extremely fast contractions. Okay? Intermediate fibers are pink fibers. They have some hemoglobin and some myoglobin. Slow fibers are red fibers. They're big leg muscles, slow contractions. So if you have a marathon runner for Olympics, they're going to be training running 20 miles a day. They're going to develop slow fibers. They're going to make them massive. They're going to make them a lot of hemoglobin, a lot of storage in there, and they're going to function for a long time. If you're a sprinter for the Olympics, 100 meters, okay, you're going to train your muscles to be fast fibers. You don't need that endurance. You don't need to build up the extras and glycogen reserve. You need the muscles to respond fast, extremely quick. And at the end of the race, they're done. Okay? So you train for different muscles can develop them. Certain people have different muscle ratios when they're born. Athletes that are faster will have fast muscle fibers. Athletes that are more in distance running will have slow muscle fibers.
So when you say that the slow fibers have more blood, right? Correct?
It still has more myoglobin, more storage.
So is that why Armstrong lost his medals? Because he was blood doping? Is that what that does?
No, blood doping gives you more red blood cells. So your oxygen content can be greater and supplied to muscles. So the muscle, any muscle will not fatigue as fast. It will affect the slow fiber muscles. It'll give them more oxygen on top of what they're only used to. In other words, when your body exists at a certain level of red blood cells, if you re-inject your own blood before the race, you get more red blood cells, you carry more oxygen. So more oxygen can deliver to those muscles, they can function longer and harder. So it makes the slow muscle fibers more effective. You don't get fatigued as easily. But yeah, that's blood doping. But you got to do it in enough time to get your body a chance to regenerate your own blood. Okay, you have 5 liters of blood, you take out a liter, you got to let your body regenerate before you reject it back in, or else it does you no good. So you got to be careful when you're doing it, and it can detect it. They can detect blood doping. Okay, so here's your slow fibers. This is what they look like. They're small diameter, they're darker color, they're more red due to the globin, the extra globin there, and they're fatigue resistant. They don't fatigue as easily. Okay, think about your leg muscles. You walk all day long, and you can keep walking. Okay, they don't fatigue. Think about your eyes. If you're reading a book and you're concentrating using your eyes, what happens to those eye muscles pretty quickly? They get tired. You want to go sleep at night? Try reading your anatomy book. Your eye muscles are concentrating, they'll get tired, and they fatigue you usually because they're fast fiber muscles, whereas your leg muscles are slow fiber muscles. And here's so you can see the difference in the histological slides. These are your fast fiber muscles. They have very large diameters. Okay? They're paler in color because they don't have as much myoglobin, and they fatigue easily. Okay? So fast fibers. Okay? Fast fibers are large in diameter, large glycogen reserve, relatively few mitochondria. Okay? You need to produce energy for the muscle to contract, and then you don't need it anymore. It's going to be a fast, quick reaction. Okay? Once it's done, it's done. It doesn't have to keep going. So you have very few mitochondria, or fewer than slow fibers. Okay? Muscles contract using anaerobic metabolism. Okay? They protect without oxygen because they don't need to keep going as long. They don't need to keep that aerobic metabolism going. They do their job and they're done. They fatigue very easily. They can contract in 0.01 seconds or less after stimulation. So your eyes can respond very quickly to a stimulus. And they do. How many times have you been driving the car and you take a turn and the sun hits you right in the windshield? What do your eyes do? Those pupils constrict extremely fast so you don't damage your retina. Okay? They're very, very fast reaction. And they produce very powerful, very quick contractions. Okay? Slow fibers are half the diameter of fast fibers. They're smaller. They take 3 times longer to contract after stimulation. You don't need them to contract that fast. They're in it for the long haul. They're not going to be the ones that contract extremely fast like that. Okay? They can contract over extended periods of time. They contain a lot of myoglobin, a lot of oxygen-containing compounds, so they have oxygen available. That gives them the red color. Okay? Muscles contract using aerobic respiration. They use oxygen. It's a slower process. It produces more ATP, it's more efficient, but it has to use oxygen and it occurs slower. Anaerobic metabolism does not use oxygen, breaks down glycogen extremely quick, but it's not very effective. You waste a lot of it, but you don't care because fast fibers they're done it, they did it, and they're done. Okay? They have very large network of capillaries, a lot of blood supply in your slow fibers because you want them to keep getting oxygen supplied, keep the contractions going. It's in it for the long haul. Okay? Intermediate fibers are between the two. That's the easiest way to explain it. It has some similarities of fast fibers and some similarities of slow fibers. They have low myoglobin, but a high concentration of glycolytic enzymes. They contract using anaerobic metabolism. Okay? But they're like slow fibers. They have lots of mitochondria. They have a greater blood supply, and they don't fatigue as easily. So they're kind of in between the fast and the slow fibers. Okay. And here's just some properties that show you the difference between the slow, intermediate, and the fast. And again, the mitochondria, you got a lot of mitochondria in the slow. You got an intermediate amount in intermediate ones and a very few in the high ones. The capillary supply is very low in the fast twitch or the fast skeletal muscle fibers, and the capillary supply is very dense or high in the slow fibers. The big thing that gives you the color difference is the slow fibers are red because of the high myoglobin content. The fast fibers are white because of the low myoglobin content. Okay. The distribution of fast fibers, okay, highly dense associated with your eye and your hand muscles, where you want things to be quick. Okay. If you take your hand and you flex it over and over and over again, it's going to fatigue very quickly. Your hands start to fatigue fast. Okay. Sprinters, like we said, Olympic sprinters that train for very quick responses. Sprinters have a high concentration of fast fibers. Repeated, intense, short, fast sprint workouts increase the fast fibers. Okay. Slow and intermediate fibers. Okay. None of these are associated with your eyes or your hands. Those are all our fast fibers. Okay. They're found in the density of your back and your leg muscles. Your back supports the main part of your body, and your leg muscles, you walk around all day. Those ones have to be in it for the long duration. Those ones have to go for a while. Marathon runners have a high amount of slow fibers. They train their muscles to go a long distance without fatiguing. Okay? And that increases the proportion of intermediate and slow fibers. Okay, any questions on fast, slow, intermediate fibers? You'll see a question like that on your midterm. So know the difference. What makes them different colors and what makes them what they are basically. All right, muscles. We can classify skeletal muscles. We're gonna name a lot of muscles in the human body. Was there a question? No, okay. We're gonna name a lot of the muscles in the human body. We have different ways of naming them. What they're attached to, how they're shaped, things like that. So muscles can be classified based on shape or the arrangement of their fibers. We can have parallel muscles, okay, where they all run the same line. We can have convergent muscles. We can have pinnate muscles. They can be unipinnate, bipinnate, or multipinnate. Okay? And we can have circular muscles. We'll show you examples of each one of those. Okay? Parallel muscles. Muscle fascicles are parallel to the long axis. Okay, so you're parallel to the long axis, like your biceps brachii and your rectus abdominis. Okay, the rectus abdominis runs through your abdomen. Those are the muscles that give you your six-pack. Everybody remember the movie 300? This is the muscle they developed, right there, the rectus abdominis. That's what they worked out. Okay, so if you look at your parallel muscle, this is your biceps. Okay, it's parallel to the long axis. Okay, and the muscles here, your rectus abdominis, your six-pack, you can see how these are all parallel Okay? The tendons that band across them. So they're long parallel muscles. Okay? So they're called parallel muscles. This is your bicep brachii and your rectus abdominis are parallel muscles. Convergent muscles. Muscle fibers from a broad area that come together. Here's your muscles, and they converge to a common point. Your pectoralis major is a convergent muscle. Here's your pectoralis muscle. Here's the base of the muscle, and it comes to a point to a tendon. It connects to bone. That's a convergent muscle. Pennate muscles. There's unipennate, bipennate, and multipennate. Pennate muscles form an oblique angle to the tendon of the muscles. So it allows the bone to pull at an angle. Unipennate, all the muscle fibers are on the same side. So in unipennate, all the muscle fibers are on the same side of the tendon, like the extensor digitorum. Extends the digits of your hands or your foot. Okay, so this is a unipennate. Here's the tendon that comes down, and all the fibers are on the same side of this tendon. So that's unipennate. And that's an extensor muscle, extensor digitorum. Extends the digits. Okay. Again, pennate muscle means oblique angle. Bipennate. Bipennate muscle fibers are on both sides of the tendon, like the rectus femoris of the femur bone. Okay. All right, here's your rectus femoris muscle. See, here's the tendon, and it's coming on both sides of the tendon. And you see the angle they're at. They're not going along the long axis. They're at an angle. They're an oblique angle. So bipennate means the muscles are on both sides of the tendon. Okay? And again, your pennate muscle is oblique angle, but multipennate means a tendon's branch within the muscle, like your deltoid. Okay? Here's your deltoid muscles. Here's the tendon, and you see these muscle fibers. Here's your cross section. You see how they branch in different sections going around the tendon. So different areas will branch. That's multipennate. So again, the unipennate is on one side of the tendon. Bipennate is on both sides of the tendon. Multipennate is when they branch within the tendon, and the tendon has muscles surrounding it, and they're run at oblique angles. Circular muscles. Muscles that form a circle. That's the tough one. Okay? They form concentric rings. They're also known as sphincter muscles. Sphincter muscles are circular muscles. The orbicularis oris, which surrounds the mouth. The orbicularis oculi, which surrounds the eye. Those are sphincter muscles. They're round muscles. You see they're circular. This is when it's contracted. This is when it's relaxed. Okay? You form the different shapes of your mouth. Orbicularis oris is the one that surrounds the mouth. Okay. You can also name them by shape. You can name them by where they're inserted, where they attach. The point of muscle attachment that remains stationary is the origin. The point of muscle attachment that is movable is the insertion. Okay. The function of the muscle depends upon how it contracts. When it contracts, it's going to shorten. And when it's connected to things, how it shortens determines its function, what it's attached to. Okay, so muscles grouped according to their primary action into 4 types. Okay, there's prime movers, which are your agonists. There's antagonists, there's synergists, and there's fixators. Okay, so your prime movers, your agonists, are responsible for producing a particular movement. Like your bicep brachii contracts and flexes your arm. That's a particular movement. That's a prime mover. Your antagonist is an action that opposes the action of the agonist. So think about your arm. Your biceps brachii contracts, okay, and your arm does this. Your triceps brachii contracts, and your arm does that. If the bicep brachii is the agonist, the tricep brachii is the antagonist. It moves them back in the opposite direction. Synergists assist the prime mover in performing an action. So it's a muscle that helps the prime mover. It's a synergist. And fixators are agonists and antagonists that contract to stabilize the joint. So if you can bend your joints like flex, like if you extend and flex, if you hold it straight, both those muscles contract at the same time to fixate this joint, to keep it from flexing and keep it from extending. So if they both contract, they cancel the other out and they keep the joint fixed. That's what fixators do. So there's 4 types. So you got agonists, antagonists, synergists, and fixators. Prime movers. Biceps brachii flexes your lower arm. And again, flexing is involved in moving the 2 bones, closing that angle, decreasing that angle. Okay. Your antagonist is your triceps brachii that extends the lower arm, increases the angle. Synergist. Synergist works in combination with the primary mover or the agonist. Okay, like the latissimus dorsi and teres contract. These 2 muscles contract to move the arm immediately over the posterior part of the body. And fixators contract at the same time to stabilize a joint, like your outstretched hand. Okay. Muscles can include their location, if you name according to where they're attached to. They can be named for their specific body region, where they're located. They can be named by their shape, their orientation, unusual features, origin and insertion, their primary function, or can represent occupation or habitual action. So basically, you pick anything you want from muscle and you can justify it. So there's no system for any muscles. Just that's how they named them. They can pick any of those they want and name muscle however they want to do it. Makes it real convenient for all of us, right? To memorize all these things. So muscles are named related to a specific region. Brachialis. Your brachialis is associated with the breaking of the arm. Tibialis anterior, associated with the anterior tibia. You can name by shape. Trapezius in the back is a trapezoid shape. Deltoid of your arm right here on the side, is the shape of a triangle, like a deltoid region of a river. Okay? So you can name them by shape. Okay, you can name by orientation, the rectus femoris muscle, the straight muscle of the leg. I swallowed that wrong. External oblique, muscle on the outside, external, oblique means it's at an angle. Okay, so looking at it, sometimes you can figure out what the muscle is, what it looks like, what it does. Biceps brachii, there's 2 origins. Teres major is a long, big, rounded muscle. They're named by origin and insertion point. Sternocleidoid mastoid muscle connects the sternum, connects the collarbone, and connects the mastoid. Okay? So it connects the sternum, the clavicle, and the mastoid from its name. Genioglossus. Genio is the attachment on your chin, the point in the anterior portion. And wherever you see the word glossus, glossus is Latin for tongue. So it connects the chin to your tongue. Primary functions, made by flexor carpi radialis. Muscle provides a flexing motion. A muscle that is near the radius, radialis, and flexes the wrist. Carpi bones make up your wrist, carpals. So it flexes muscles of the wrist by the radius. Okay. Abductor longus. Adductor means to bring towards the midline. So you bring your motion in towards the midline. And this is an adductor longus. It's a long muscle that adducts the leg, pulls it in towards the midline. Okay, so most of the time contractions, the muscle causes action. When you contract a muscle, something usually happens. This action is applied to a lever. The lever is a bone. Lever moves a fixed point called your fulcrum. That's your joint. That's your fulcrum point. The action of the lever is opposed by force that acts in the opposite direction, whether it's gravity or something else. You're acting against another force. That's what keeps your arm in motion. There's 3 classes of levers. There's first class, second class, and third class. Okay, first class. The fulcrum lies between the force and the resistance force, like tilting your head forwards and backwards in the yes motion. Okay, so that's a, oops, wait, I went too far backwards. Go back. All right. So tilting your head forward and back is a first class lever action. Second class lever action. The resistance located between the applied force and the fulcrum or the joint, like standing on your tiptoes. You roll yourself up. That's a second class lever or action. Third class, the force is applied between the resistance and the fulcrum point. Example is flexing your lower arm. It's a third class type of lever action. Sometimes you have pulley systems. We use it all the time when we do mechanic work. When we take an engine out of a car or motorcycle, use a pulley system to lift it up. Makes it a lot easier. Okay? So sometimes the tendon may loop around a bony projection. Bony projection is a pulley system, like the lateral malleus of the ankle and the trochlea of the eye. Okay? It's a very effective way of moving your eye muscles. It's a pulley system. As a tendon contracts, the muscle pulls just along that edge. Okay? So here's your pulley system. This is your pulley. And as this muscle contracts, it pulls over that bone and causes a certain action. So the patella is another example of an anatomical pulley. Okay? It swings over the patella, and it's used as a pulley system for extension of the leg. As this comes up, this muscle is pulled here, it goes over the patella, and this is pulled here, just like a pulley system, just like you would use in your garage. Okay? Aging in the muscular system. As you get older, things change. Everything changes, not just muscles, but change occur muscles as you age, right? From 21 on, it's all downhill, guys. So skeletal muscle fibers become smaller in diameter. Okay? Due to a decreased number of myofibrils. You are not decreasing the muscle cells. You decrease the number of myofibrils, the actin and myosin inside of them. They contain less glycogen reserve. You can't use your muscles as long, and they contain less myoglobin. They don't have enough energy that keeps them going. All the above, okay, all the above causes a decrease in strength and a decrease in endurance. And your muscles tend to fatigue very rapidly as you get older. Okay. There's also a decrease in the myosatellite cells. So even though it must have very limited repair, you get less as you get older. There's an increase in fibrous connective tissue. So your muscle tissue gets replaced with fibrous connective tissue. Okay? This is due to the process of fibrosis and the ability to recover from muscle injuries decreases tremendously as you age.